Welcome, everyone, and Happy New Year. I'm Rita Valentino. I'm the Director of the Division of Neuroscience and Behavior at the National Institute on Drug Abuse. And today, it's my honor to introduce the first Director's Wednesday afternoon lecture series of 2024. And you're in for a treat today. Today's speaker is Dr. Leigh Monje. Michelle is the Myelin Professor of Pediatric Neuro-oncology in the Department of Neurology and Neurologic Sciences at Stanford University, and she's a Howard Hughes Medical Institute investigator. She received her MD and PhD in neuroscience from Stanford and completed her residency training in neurology at Mass General Brigham. She then went on to back to Stanford for a clinical fellowship in pediatric neuro-oncology. Dr. Monje is truly a scientific pioneer whose research integrates neuroscience, immunology, and cancer biology with an emphasis on neuro-glial communication. Her laboratory has made several impactful discoveries, including the discovery that neurons and glia can communicate through functional synapses, and that neuronal activity promotes the progression of malignant gliomas through this communication. She identified molecules that drive the growth of gliomas and that may be targets for treatment. So her basic research really is translating to treatments for devastating brain tumors in children, and several of her discoveries have led to clinical trials. Her work has been recognized with numerous honors, including an NIH Director's Pioneer Award, a MacArthur Fellowship, election to the National Academy of Medicine, the 2023 Paul Marks Prize, and the 2023 Richard Lounsbury Award from the National Academy of Sciences. Before I hand it over to our speaker, I want to remind you that today's CME credit code is 50107. And also, to submit questions, please click on the Send Live Feed button to submit. So thank you, and now I'll hand it over to Michelle.
It's so wonderful to be joining you. Thank you so much for the invitation to speak with you today. These are my disclosures. Broadly speaking, my laboratory tries to understand the molecular language that cells use as they work together to build and remodel the brain. And I want to tell you one story today in two parts about the way in which neurons, in an activity-dependent manner, communicate with the glial cells that form the myelin sheath, and how we're beginning to understand that these interactions are really dynamic and adaptive, and that these activity-dependent, even relatively subtle changes in the myelinated infrastructure of the nervous system can tune circuit function relevant to various forms of cognition and behavior. And then how these really powerful interactions between neurons and oligodendroglial cells are hijacked in the context of glial malignancies. But I like to start every talk with a pretty broad consideration of neural development, and I think the best way to do that is to show you pictures of my children. So this is my daughter Sophie when she was an infant, and we just appreciated that there was so much neurodevelopment that human infants undergo every day in the first year of life that we needed to give her a way to keep up with that, to keep track of it. So we made her kind of a neurodevelopmental to-do checklist, and we put it on her onesie because we didn't know where else to put it. And it really is astounding when you consider all of the neurodevelopment that human infants have to accomplish every single day in the first year of life. For example, humans make something on the order of 500 billion new synapses every day in the first year of life. 14,000 new hippocampal neurons daily, 127 million new cerebellar granule neurons, the cerebellum, having a major component of postnatal development. But perhaps, as impressive as that all is, perhaps the most onerous task that humans have in the postnatal period is to myelinate their central nervous system. Myelin is chiefly a postnatal developmental process. At birth, there's very little myelin in the human brain. It begins just before birth, myelination, around the center of the brain, around the central sulcus. And then there's a massive wave of developmental myelination progressing from the sort of central focus towards the poles of the brain and beginning in the cervical spinal cord and progressing down towards the lumbosacral spinal cord. But after this massive wave of myelination, it doesn't end there. This is a very protracted developmental process spanning about 30 years of human life. And as protracted and long as that is, there are some recognizable patterns of developmental myelination. The progression of developmental myelin follows really predictable topographical as well as chronological patterns happening in certain parts of the nervous system at certain ages. So, for example, more basic circuitry tends to myelinate prior to more complex circuitry. And as an example of that, there's a discrete wave of developmental myelination that occurs in the ventral brainstem, in the corticospinal tracts during mid-childhood, around ages 6 to 8. And then a different, distinct wave of myelination in the neocortex and intercortical association fibers during adolescence and young adulthood. And so myelination is the process by which oligodendrocyte precursor cells generate new oligodendrocytes that ensheath the axonal membrane to decrease transverse capacitance and increase the speed of neural impulse conduction. It doesn't end at the end of this 30-year period of development, but actually in some regions of the nervous system, like the neocortex and its projections, myelination continues throughout life. And there's an accumulation of myelin, as you can see in this fairly old, actually, historical lithograph of the human neocortex throughout the lifespan. And this increase in the number of oligodendrocytes and increase in myelination of the neocortex has been, also seen in rodent models and visualized through live imaging. And this is, I think, intrinsically really exciting and interesting, but it's also very important to understand because glial malignancies of childhood, the most common solid brain tumor of child, the most common solid tumor of childhood are brain tumors, and gliomas are the most common form of childhood brain cancer. These also arise in a pretty predictable spatiotemporal pattern that maps pretty well onto that spatiotemporal pattern of developmental myelination, such that at a time when there is this discrete wave of myelination in the ventral brainstem during mid-childhood around ages 6 to 8, this is the peak incidence of one of the worst human malignancies, diffuse intrinsic pontine glioma. And similarly, when there is that discrete wave of neocortical myelination in adolescence and young adulthood, this is when hemispheric high-grade gliomas like glioblastoma tend to happen in adolescents and young adults. And this pattern is concordant with observations from my laboratory and from many others that many forms of glioma appear to arise from and very closely molecularly resemble these myelin-forming precursor cells, oligodendrocyte precursors. And so that begs a very basic question. What regulates the proliferation and functional differentiation of these oligodendroglial lineage precursor cells? Well, one idea that's been in the literature for some time, first introduced by Ben Barris in the early 90s when he was a postdoctoral fellow with Martin Raff, and then later supported by some really beautiful work, much of it done in Doug Fields' lab at NIH, as well as some correlational work in human neuroimaging by Heidi Johansen-Berg, suggested that neurons themselves may be regulating the extent to which their axons are myelinated. And this was a very compelling idea. It was also, a decade ago, a fairly controversial idea because we know that there are also activity-independent modes of myelination, that oligodendrocytes in a petri dish with any inner nanofiber of the right diameter will myelinate that fiber as long as it's the right size. So there's clearly activity-independent modes of myelination, and the question was, in awake behaving animals, in the intact nervous system, is myelin plastic and adaptable? Does neuronal activity, at least in some regions, in some circuits, at certain ages, regulate the myelinated infrastructure of a circuit? And so when I started my laboratory now, about 12 years ago, this was one of the first questions that we were really excited to ask. And so I want to focus the first half or so of the talk on this idea of myelin plasticity, of activity-dependent changes in oligodendroglial lineage cells and in myelin structure. And to introduce you to the people in my laboratory who've really led this work, this was a project that was initially started by my very first postdoctoral fellow, Erin Gibson. Erin is absolutely brilliant, went on to start her own laboratory in the Department of Psychiatry at Stanford and is continuing to do really important oligodendroglial myelin-related work. This is Anna Garrity, who was a postdoctoral fellow with me, and I was lucky enough to keep in my lab as a senior staff scientist. And she continues to ask questions about activity-dependent regulation of myelin-forming cells. Juliette Knowles was a postdoc with me. She is now an independent assistant professor in the Department of Neurology and a pediatric epileptologist. And this is Belgin Yalcin, who's just finishing her postdoc and is on the job market right now. And what Erin and Anna, Juliette, Belgin, and many others in the lab have done is to leverage techniques in modern neuroscience to try to ask these questions about neuron interactions with other cell types. And so, as I think many people in this audience know well, in vivo optogenetics has been a really transformative technique for neuroscience, and we leveraged it here to try to understand neuron-glial interactions. And as many people know, in vivo optogenetics allows for control of targeted populations of neurons or other cell types that are engineered to express these light-sensitive opsins, light-sensitive ion channels. This is channel rhodopsin 2. It's a blue light-sensitive cation channel. And if we express channel rhodopsin 2 in cortical projection neurons and then deliver blue light over the surface of the motor planning area, or M2 cortex, and we pulse that light at the frequency at which these cortical projection neurons typically fire, then we can evoke physiomimetic activity within the circuit. And we know that we've done that if we induce a complex motor output. So here's a mouse in a cage, and complex motor output for mice in cages often looks like walking in a circle. So this mouse is having 20 hertz optogenetic stimulation of glutamatergic cortical projection neurons in the motor planning area and begins to walk in a circle. And that lets us know that we've stimulated that circuit successfully and can begin to ask other questions about how other cell types within the stimulated circuit respond to changes in neuronal activity. And what we discovered now a number of years ago is that cortical projection neuronal activity, and really quite specifically cortico-callosal, but not corticofugal neuronal activity, elicits a rapid and robust proliferative response of oligodendrocyte precursor cells, specifically within that cortico-callosal circuit. If we fate map those cells over time by administering the thymidine analog EDU to the mice at the same time that we do that optogenetic stimulation, then we find that the proliferating oligodendrocytes generate, proliferating oligodendrocyte precursor cells generate new oligodendrocytes, and that the myelinated ultrastructure of that circuit changes. And it changes in a way that we would predict might alter circuit dynamics and influence function. And indeed, what we found is these mice exhibited improvement in motor functional performance that depended upon the generation of new oligodendrocytes. So we wanted to understand the molecular mechanisms mediating these neuron oligodendroglial interactions, and we hypothesized that activity-dependent neurotrophin signaling, specifically activity-dependent BDNF signaling to its receptor TrkB on oligodendrocyte precursors, might mediate some of these interactions or might be important for this mechanism. And so we tested that idea using two different genetically engineered mouse models. In the first mouse model made by Mike Greenberg's lab at Boston Children's, there are normal baseline levels of BDNF, but the BDNF gene has been engineered to delete the CREB binding site in promoter 4, so there's no activity-dependent increase in BDNF expression or secretion. And in a second mouse model, rather than modulating the BDNF ligand, instead we specifically and conditionally deleted the BDNF receptor TrkB just from oligodendrocyte precursor cells in young adulthood. And what we found is that if we repeated that experiment and optogenetically stimulated cortical projection neuronal activity in either of these genetically engineered mouse models, the activity-regulated response was entirely lost. In contrast to BDNF wild type or TrkB wild type mice, in which you see this expected increase in OPC proliferation in response to cortical projection neuronal activity, in mice that either lack activity regulated BDNF changes or in mice whose OPCs have been engineered to lack the TrkB receptor, there is no increase in OPC proliferation, no oligodendrogenesis, and accordingly no changes in myelin in these mice. And so while we think that the mechanism mediating myelin plasticity is much more complex than just this one signaling interaction, what we've found is that because that BDNF to TrkB signaling interaction is required for the activity-dependent changes, we can use it as kind of a molecular handle to begin to understand the role of myelin plasticity in various behaviors. And using this approach, we and others using similar approaches have found that neuronal activity evoked either optogenetically, chemogenetically, or by changes in experience play really important roles in a range of different brain functions, including motor function, motor learning, spatial learning, fear learning, attention, memory, and coding. Now, the changes in myelin take a couple of different flavors. I've talked so far about de novo myelination, which includes OPC proliferation, the generation of new oligodendrocytes, and then the generation of new myelin internodes on previously unmyelinated axons or previously unmyelinated regions of axons. In addition to de novo myelination, there's also myelin remodeling, changes in existing oligodendrocyte generated myelin internodes such that activity regulates the geometry of these internodes. And those nodes can become either longer or shorter, thicker or thinner. And these activity-dependent remodeling changes can subtly change conduction velocity and influence circuit dynamics. We and others have been trying to understand the molecular mechanisms that mediate these interactions. And as I mentioned, there are some paracrine factor interactions. I talked about BDNF signaling from cortical projection neurons. Charles French-Constant's group has found that in the superficial cortex that endothelin signaling is an important factor mediating myelin remodeling. Different molecular mechanisms may be at play in different neuronal subtypes and in different circuits. But in addition to these paracrine factors, there are also bona fide synapses that form between neurons and oligodendrocyte precursor cells. These neuron to OPC synapses were first described by Dwight Burgles in 2000, so 24 years ago. And they have been very well characterized. They're both glutamatergic and GABAergic neuron to OPC synapses. The functional role of these synapses in myelin biology is still really not well understood. They certainly exist. They exist on the majority of OPCs, but what they do is something that's still very much a topic of intense investigation. What is clear is that these, even relatively small changes in myelin structure can have really important effects on circuit dynamics. And the principle that has emerged now, both from computational work as well as experimental work, is that in the healthy nervous system, these small myelin changes tune circuit dynamics to promote coordinated circuit function. For example, by promoting, synchronous oscillations between nodes within a neural network. And so if myelin plasticity, is increasingly recognized as playing important roles in various cognitive functions, such as attention, memory, and learning, it would make sense that loss of myelin plasticity might be important in diseases of impaired cognitive function. And two diseases that we have studied, and I'll tell you some vignettes about, include cancer therapy-related cognitive impairment. As a neuro-oncologist, especially as a pediatric neuro-oncologist, this is a disease that my laboratory has spent many, many years studying and that we think a lot about. And as I'll share with you, the mechanisms at play in disrupting myelin plasticity in the context of cancer therapies are immunological in nature. And so we worried that there might be a similar pathophysiology playing out in profoundly inflammatory syndromes like COVID, and that might contribute to COVID brain fog. And so we've studied that as well. And I'll tell you a couple of vignettes about the importance of myelin and myelin plasticity in diseases of impaired cognitive function. So after, as I think many in this audience know well, after cancer therapies, there is a reproducible syndrome of cognitive impairment characterized by impaired attention, memory, speed of information processing, and multitasking. Using a mouse model of high-dose systemic methotrexate exposure, we found that there was a direct effect of methotrexate on microglia, the resident myeloid cells of the nervous system, and more specifically on microglia in axon tracts, just the microglia in white matter, which are our unique subpopulation of microglia. The systemic methotrexate exposure activates these and causes reactivity in these white matter or axon tract microglia. Those microglia, once reactive, then consequently cause neurotoxic astrocyte reactivity, and together these reactive glial cells dysregulate both myelin homeostasis as well as plasticity. And we know that the microglial reactivity is central to this pathophysiology because if we transiently deplete microglia, in this case using a CSF1R inhibitor, that restores myelination and rescues cognition in this mouse model. When the pandemic began, already understanding that neuroinflammation and microglial reactivity played a central role in cancer therapy-related cognitive impairment, I was very worried that we were going to begin to see a similar brain fog syndrome. And very quickly in the pandemic, it became clear that many people, even after relatively mild COVID, were suffering from a syndrome that looked for all the world like cancer therapy-related cognitive impairment, also characterized by impaired attention, impaired memory, impaired speed of information processing, and multitasking. And so I reached out to Abhinath at NINDS and also to Akiko Iwasaki to collaborate and try to test this hypothesis that even relatively mild respiratory restricted COVID, in the absence of any brain infection, might cause a similar dysregulation of neuron-glial interactions. And so we used a mouse model that Akiko Iwasaki's lab had developed in which there is a relatively mild COVID induced in mice, and it is engineered to be restricted to the respiratory system. So only the respiratory system expresses human ACE2. And so when mice are inoculated intranasally with SARS-CoV-2, the infection is restricted to the respiratory syndrome. The mice don't get very sick. They don't lose much weight. They clear the virus from the respiratory system within about 7 days. And yet when we look at the brain, either 7 days later or 7 weeks later, I'm showing you here the 7-week data, there is a similar pattern of persistent microglial reactivity just in axon tracts, just in the white. When we look at the myelin then, after this mild respiratory exposure, both at 7 days in this top row and at 7 weeks in this bottom row, we see that there is a loss of myelinated axons. There's a loss of oligodendrocytes as well to explain that. And that when we compare that loss of myelinated axons that we see after mild respiratory restricted COVID and after chemotherapy, we see a very similar decrease in myelinated axon numbers. Okay, so we think that myeloplasticity plays numerous roles in healthy cognition and that impairment of myelin homeostasis and plasticity can contribute to cognitive impairment. What happens if there's too much myelination in diseases characterized by aberrantly increased neuronal activity or aberrantly increased patterns of activity? And we've studied this idea that this activity-dependent myelination might instead be maladaptive in the context of generalized epilepsy syndromes, and more recently in some unpublished work I'm going to share in reward learning in response to drugs of abuse. So I'm going to tell you 2 more vignettes about the role of aberrantly increased maladaptive myelination. So Juliette Knowles, when she joined my lab, wanted to understand whether activity-dependent myelination actually might be playing a pathophysiological role in generalized epilepsy syndromes. So this is work that we did together with John Huguenard here at Stanford, using his mouse models of absence epilepsy, in which we found that indeed after the onset of epilepsy in both a rat model and here in a genetically engineered mouse model of generalized absence-like epilepsy syndromes, after the onset of the seizures, there is an increase in both the number of myelinated axons as well as the thickness of the myelin sheet. This is not evident at early ages before the seizures begin. So this is we think, due to the seizures. Indeed, we find these myelin changes only within the seizure network. If we look outside of the seizure network, in this case, a thalamocortical network, we see no such myelin changes. And these myelin changes depend on the seizures. If we effectively treat these seizures using, in this case, ethosuximide as an anti-epileptic drug, then these myelin changes never happen. And so the question was, then, does the aberrantly increased myelin, which we would predict would increase oscillatory synchrony between nodes in the seizure network, does that contribute to the increase in seizure severity that is seen as part of the natural history of generalized epilepsy syndromes? And so to test that, we leveraged our new understanding of the role that TrkB plays in activity, the necessary role that TrkB plays in myelin plasticity. And we looked at mice that, we had engineered to not have myelin plasticity. So we looked at wild-type mice with this genetic syndrome inducing generalized epilepsy, and then in mice in which we conditionally and inducibly delete TrkB just from OPC so that they can't respond to the activity-dependent signal. And what we found when we did intracranial EEG in these mice was that the ictal coherence, the synchrony between nodes within the seizure network, was decreased in the mice that lacked myelin plasticity. And then looking over time, we found that mice that lack the ability to respond to these activity-dependent signals did not have the increase in seizure severity that occurs in mice with intact myelin plasticity. There were far fewer seizures happening daily late in the course of the disease in mice that lack myelin plasticity compared to those with intact myelin plasticity. In more recent work, Belgin-Yeltsin in my lab has wondered whether there are activity-dependent changes in dopaminergic reward circuitry that could be triggered by drugs of abuse, and whether that contributes to the reward learning that occurs. And so to test this idea, we first did optogenetic stimulation in the ventral tegmental area of dopaminergic neurons. We also did GABAergic neuron optogenetic inhibition, which in turn upregulates dopaminergic neuronal activity, just to look at this in a couple of different ways, using a couple of different optogenetic strategies. And what we found was that dopaminergic neuronal activity in the ventral tegmental area results in new oligodendrogenesis that's restricted to the VTA. We don't see it in other parts of the reward circuitry. And when we look by electron microscopy at the myelin, we find that there is an increase in both the number of myelinated axons in VTA as well as the thickness of the myelin sheath, and that these myelin changes are restricted to the proximal portion of dopaminergic axons. When we, instead of optogenetically stimulating these mice, increase dopaminergic neuronal activity by giving drugs of abuse, when we administer either morphine or cocaine, we see a similar oligodendrogliogenesis response, new oligodendrocytes made, again, specifically within the ventral tegmental area. And of course, as expected, this goes along with conditioned preference, the conditioned place preference that the mice exhibit to the chambers in which they receive the drug. So all the mice that received morphine or cocaine have a conditioned place preference, and associated with that conditioned place preference is this increase in oligodendrogenesis. So does the oligodendrogenesis have anything to do with that reward learning? Well, if we prevent oligodendrogenesis, either by using that conditional TrkB oligodendrocyte-specific knockout of TrkB model, or using a similar strategy in which transcription factor required for oligodendrogenesis, MRF, is conditionally deleted. In either of those genetic mouse models that lack the ability to make new oligodendrocytes, when we administer morphine, we find that there is a loss of the conditioned place preference. The mice don't learn which chamber they receive the morphine in. And we can understand this if we look at dopamine release dynamics in nucleus accumbens. So the ventral tegmental area, dopaminergic neurons project here to the nucleus accumbens. And in mice that lack the ability to make new oligodendrocytes during the learning process, They have really disordered dopamine release, here shown in red, compared to mice with intact myelin plasticity, who have this expected peak in dopamine release when they enter the chamber in which the drug of abuse was administered. Okay, so we think that myelin plasticity is important, that it contributes to attention, memory, and various forms of learning, that loss of myelin plasticity contributes to diseases of impaired neurological function, evident in cancer therapy-related cognitive impairment and in the COVID cognitive impairment that happens as part of the long COVID syndrome. And that this myelin plasticity, if it's really important in regulating circuit dynamics, can become maladaptive if that is dysregulated. And that we see play out in generalized epilepsy, and we see it contribute to learning in the context of drugs of abuse. But what happens if the oligodendrocyte precursor cell has an oncogenic mutation? What if this healthy oligodendrocyte precursor cell instead is a glial cancer cell? Could these really powerful interactions between neurons and glial cells be subverted and hijacked instead of promoting adaptive myelination to promote cancer growth? And so I'd like to focus the second half of the talk on the idea of malignant myelin plasticity, on activity-dependent growth of glial malignancies. And to introduce you to the people from the lab who have really led this work, this is Hamza Venkatesh, who began the project in the lab when she was a absolutely brilliant cancer biology PhD student. She stayed with me for her postdoc and is now leading her own independent lab at Dana-Farber Cancer Institute, Brigham and Women's Hospital. She is wonderful. If there's anybody out there in the audience looking for a postdoc, I cannot more strongly recommend Hamza. She's absolutely, absolutely brilliant. Similarly brilliant, and similarly someplace you should go for a postdoc, if there's anyone in the audience looking for such a position, is Yuan Pan. She was a postdoc with me together with David Gutman at WashU, one of my very close collaborators. And after finishing up, she started her own laboratory at MD Anderson Cancer Center. Katie Taylor and Tara Barron are presently postdocs in the lab, and Katie is on the job market this year. And as a pediatric neuro-oncologist and someone focused on understanding the biology of gliomas, these are the tumors that I spend the most time thinking about in my laboratory studies. As I mentioned before, gliomas of childhood arise in a pretty predictable spatiotemporal pattern, to the extent that if you tell me a child has a glioma and give me their age, I can probably tell you where it is. These are the diseases, examples that I'm going to present in the coming slides. And I just want to again highlight that this spatiotemporal emergence of these gliomas really speaks to a developmental process that has gone awry. This is an optic pathway low-grade glioma.
Transferred over to me, perhaps. I sense that might have been the case.
Chris,
yeah.
I'm not sure if
I'm in the middle of hosting a lecture right now. I picked up the phone because I thought it might have been No, you wouldn't have known.
Chris, you need to mute.
Okay, he's muted now. So this is NF1 associated optic pathway glioma. These tend to occur in relatively young children and very commonly in association with NF1. They also can happen sporadically. As I mentioned earlier, this is diffuse intrinsic pontine glioma, also called pontine location diffuse midline glioma. This is one of the worst imaginable human malignancies. This is a universally fatal cancer that tends to occur in school-aged children, but can occur at any age. A very molecularly related disease is diffuse midline glioma of the thalamus. This tends to happen in slightly older kids. And then hemispheric high-grade gliomas tend to occur in adolescents and young adults. And the idea that neurons might be playing an important role in the biology of these tumors was something that actually a really brilliant neuropathologist recognized about 100 years ago. H.J. Shear in his description of what he called the secondary structures of Shear and what we now call perineuronal satellocytosis, the propensity of malignant glioma cells. I'm just going to show you a cartoon version of what you're seeing here. Malignant glioma cells here depicted in green tend to cluster in very tight microanatomical association around mature neurons in the tumor microenvironment and then invade along axon tracts. And this really close microanatomical association suggests that there might be a very important signaling relationship between these two cell types. And that was something that back when I was a pediatric neuro-oncology fellow and when I was a neurology resident, I spent a lot of time thinking about looking at, histopathological samples from my own patients. And here's an example of perineuronal satellocytosis in the ventral pons of a 5-year-old child with diffuse intrinsic pontine glioma, DIPG. And looking at this, it really struck me that the interaction between these two cell types should be studied. I also want to just acknowledge the incredible contribution that patients and their families make to research in general and to the studies I'm about to show you. This particular H&E image comes from this 5-year-old patient that I cared for when I was the fellow, and it was his early postmortem tumor and brain donation that enabled us to make the very first cell culture and xenograft mouse model of DIPG. And it's through similar such donations from patients and their families that we and others are able to do much of the work that we do. So I just want to acknowledge that incredibly important contribution. And so to ask whether neuronal activity similarly affects malignant glioma cells as it does their normal glial precursor counterparts, we did that same optogenetic experiment, optogenetically stimulating cortical projection neurons, but this time in the context of a diffusely infiltrating cortical glioblastoma isolated from an adolescent patient of mine. And what we found was that when we optogenetically stimulated these cortical projection neurons, that indeed, specifically within the stimulated circuit, there was an increase in the proliferation rate of the tumor cells, and that resulted in an overall increase in tumor burden, specifically within the stimulated circuit. So brain activity can influence brain cancer growth. Later, and in collaboration with David Gutman's lab, we looked at low-grade gliomas occurring in an NF1 optic pathway glioma mouse model, and found that if we optogenetically stimulated the optic nerve prior to the time of tumor onset, which very reliably in this model occurs at 9 weeks of age, that the tumors that emerged, shown here with the black dots, were much larger than those that occurred in litter-mate controls that were identically manipulated, but in whom we just simply did not turn on the blue light. So we see now a role for neuronal activity regulating the growth of both high-grade and low-grade gliomas. Now, that optic pathway glioma mouse model was a really exciting opportunity to ask questions not only about glioma growth, but also because we know exactly where and when these tumors are going to emerge, we can ask questions about tumor initiation. And so we wanted to modulate activity in the optic nerve. And, optogenetics is a lot of fun to use, and we know, it's very effective, but in the optic nerve, we don't need to use optogenetics. We can simply modulate optic nerve activity by modulating visual experience. And so we tried to get the mice to wear these sunglasses, and that was a disaster. There was plastic everywhere. I don't recommend it. On Zoom, I never know if my jokes land, but anyway, I'm kidding. We didn't actually do that. Instead, we just put mice in complete darkness. And we did that at various time points around the time of tumor initiation. So as I mentioned, these tumors always occur at 9 weeks of age. They happen in more than 95% of the mice. By 9 weeks of age, there's a tumor in the optic nerve. If we decrease optic nerve activity by dark rearing the mice, either beginning at the time of tumor initiation at 9 weeks or just after tumor initiation at 12 weeks, when we examine the mice at 16 weeks, we find that there are far fewer and much smaller tumors compared to litter-mate controls that simply were raised with normal visual experience. And if instead we dark rear the mice after optic nerve and optic pathway development, but prior to tumor onset, starting at 6 weeks of age, then absolutely no tumors form, even if we replace the mice into normal light cycles, normal visual experience after what appears to be a critical susceptibility period for tumorigenesis. And this is in comparison to 100% tumor incidence in mice, littermate control mice, simply raised with normal visual experience. So there's some really powerful interactions at play here. And we wanted to understand the mechanisms, of course, that mediate these effects of neurons in an activity-dependent way on glioma cells. And so we first hypothesized that there may be activity-dependent paracrine signaling, activity-dependent growth factors, essentially, released as a result of action potentials. And so we tested that idea using an explant model. We took explants of either mouse cortex or, in the case of low-grade glioma, mouse retina and optic nerve. And then we collected the secreted factors into conditioned medium from around these explants when the neurons in the explants were at varying levels of neuronal activity. So they were either tetrodotoxin silenced, spontaneously active, or optogenetically stimulated. And then when we took this conditioned medium and placed it onto cultures of glioma cells, we found that there was an activity dose-dependent increase in glioma proliferation lost if the neurons in the explants had been silenced with tetrodotoxin. Now, as many people in the audience know, there are many forms of molecularly and clinically distinct glioma, but we found that the response to these activity-regulated secreted factors was conserved across multiple forms of both high and low-grade glioma from kids and from adults, including IDH wild-type pediatric hemispheric glioblastoma, IDH wild-type adult hemispheric glioblastoma, histone mutant DIPG pontine diffuse midline glioma, IDH mutant anaplastic oligodendroglioma, and NF1 mutant optic glioma. And so now the question becomes, what are the factors that are secreted into this conditioned medium? Summarizing a couple years of biochemistry and proteomic work in a single text box, as we do, we identified two key activity-regulated secreted factors. Not surprisingly, just as it plays a key role in many neurodevelopmental contexts and also in promoting myelin plasticity, we found that brain-derived neurotrophic factor was one important activity-regulated secreted mitogen. But really unexpectedly, we found this molecule. We found a shed form of neuroligin-3. Now, neuroligin-3 is a really well-known, very important synaptic adhesion molecule, first discovered by Thomas Sudoff. It plays very important roles in regulating synaptic strength at both glutamatergic and GABAergic synapses, but it was not known to be a mitogen in any context, and actually it wasn't even known to be secreted. But we find that neuroligin-3 is cleaved at the membrane, releasing this large N-terminal ectodomain in a strictly activity-dependent manner through the enzymatic activity of the metalloprotease ADAM10. So the next question is, what cell types are shedding neuroligin-3? And so we looked at the gene expression profiles of different neural cells and found that in addition to neurons, which of course express neuroligin-3, there was also very high expression by glial cells, and especially by oligodendrocyte precursor cells. And so we did some genetic mouse modeling experiments in which we conditionally deleted neuroligin-3 from various subtypes of cells, either from subtypes of neurons or from oligodendrocyte precursors, and found that actually OPCs were the chief source of shed neuroligin-3, which places the OPC in the tumor microenvironment in a really important way, and also asks some very important basic questions about the role of neuroligin-3 in myelin biology, something that is the topic of another talk, but we've been following up in a very focused way. So we then wondered, how important is this mechanism? There are lots of cell intrinsic mechanisms that mediate glioma growth and many microenvironmental factors. How important is this one molecule? So to test that, we xenografted brain cancer cells into the environment of either the neuroligin-3 wild type or neuroligin-3 knockout brain. And what we found was really surprising. Rather than just slowing in growth, we found that tumors utterly stagnated in the absence of neuroligin-3 from the brain microenvironment. In contrast to these GFP-labeled glioma xenografts growing and invading across the corpus callosum that we see in mice with wild-type neuroligin-3, and those that lack neuroligin-3 in the brain, these high-grade glioma cells simply stagnate. And we can see this in real time using in vivo bioluminescent imaging of the tumor burden. And a snapshot of that here shows you that in contrast to the growth of tumors, here's a 5 to 10-fold change in tumor burden over several months in wild type brain. In the absence of neuroligin-3, the tumors really had not grown. And this apparent dependency on neuroligin-3 was conserved across multiple different forms of glioma and in multiple different brain regions, but did not extend to a patient-derived model of breast cancer brain metastasis, suggesting that while neuroligin-3 may be important for many forms of glioma, it is not necessarily important for all cancers growing in brain. So then we turned back to that optic pathway glioma model, and we wondered whether neuroligin-3 was a mechanism contributing to this really powerful effect of optic nerve activity on optic glioma growth. And we wondered that not only because of the important role that we saw playing out in high-grade gliomas, but also because we noticed that in the NF1 optic nerve, remember, this is a tumor predisposition syndrome in which all cells in the body have lost one copy of functional neurofibromin 1, that there was an aberrant increase in neuroligin-3 shedding. And so we wondered whether that might actually represent part of the susceptibility to tumors in that structure. And we did a very similar kind of experiment. In this case, we crossed a neuroligin-3 knockout mouse to the NF1 optic pathway glioma mouse model that the Gutman lab had generated. And what we found was that the loss of neuroligin-3 in this model resulted in far fewer and much smaller optic nerves, even with normal visual experience, suggesting that neuroligin-3 is a really important mechanism here. And concordant with that, when we treated the NF1 optic pathway glioma mice with ADAM10 inhibitor, that also starkly decreased tumor growth and initiation. So what is going on with neuroligin-3? We know that neuroligin-3 is shed in this activity-dependent manner through the enzymatic activity of ADAM10, that it then binds to the glioma cell, and we're working hard and perhaps have just identified the binding partner in glioma. It's not the canonical binding partner of neuroligin-3. It's not orexin. And what we do know, though, is that downstream, there is a stimulation of numerous oncogenic signaling pathways, including early upstream stimulation of focal adhesion kinase, and then downstream SRC, RAS, and PI3 kinase mTOR pathway. So that helps us understand its roles in mitogen. It helps explain the sufficiency of neuroligin-3 in promoting glioma proliferation, but it really doesn't explain that unexpected dependency. So digging deeper and looking at the gene expression changes in response to neuroligin-3, we found that there were a number of synapse-related genes that are regulated by neuroligin-3 binding to the glioma cell. And in fact, a number of very interesting processes, genes relevant to very interesting processes are regulated by neuroligin-3 binding in the glioma cell. So in addition to these synapse-associated genes, including a feed-forward effect of neuroligin-3 on its own expression, upregulation of the gene encoding the BDNF receptor and TREC2, TREC-B. There were a number of glutamate receptor subunits, especially AMPA receptor subunits, and other synapse-associated structural proteins. We also found that neuroligin-3 maybe acting as sort of a master regulator of gene expression, regulating synaptogenic factor expression of structure that I'll explain in a few slides called tumor microtubes, regulation of other ion channels like potassium channels and axon guidance molecules. But focusing to begin with on these synapse-associated genes, that triggered for us the thought, the seemingly crazy thought at the time, that like there are synapses between neurons and healthy glial precursor cells, there might also be synapses between neurons and glioma cells. And indeed, when we look by immunoelectron microscopy, in which we can unambiguously identify the malignant cell by immunogold labeling, we see these really clear synaptic structures with presynaptic neurons and postsynaptic glioma cells. Testing the idea that neuroligin-3 may be functioning in perhaps its most fundamental role to promote the formation of these synapses, we find far fewer synapses in the absence of neuroligin-3 from the tumor microenvironment. But are these neuron-to-glioma synapses just a shadow of the cell type from which these tumors emerge, or might they actually be electrophysiologically functional? To test this, we did whole cell patch clamp electrophysiology, patching onto these GFP-labeled tumor cells xenografted into a really experimentally tractable circuit in the hippocampus. And then after we recorded from the glioma cell, we can dye fill it with Biosite just to confirm that we weren't in fact reporting from a tumor cell and not accidentally from a normal cell. And what we found when we did this experiment is that there were very clear excitatory postsynaptic currents in a subset of the tumor cells in every tumor model that we examined. These depend upon action potentials. They're blocked by tetrodotoxin. They express multiple electrophysiological characteristics of bona fide synapses, including paired pulse facilitation and single vesicle events, many EPSCs. And this first kind of synapse that we identified are mediated by AMPA receptors, and more specifically by calcium permeable AMPA receptors. This was such a crazy set of findings that reassuringly, it completely simultaneously, Frank Winkler's group and together with Thomas Kunar in Heidelberg discovered the same thing. We co-published those papers, and it was really nice to have validation of this really different way of thinking about the cancers, at the time of publication. Now, in the healthy brain, AMPA-dependent synapses exhibit mechanisms of adaptive plasticity, and we wondered whether that might be yet another aspect of neurophysiology that the tumors are hijacking. And there are a few different, classical mechanisms of neuroplasticity based on the gene expression profiles. We thought that BDNF-mediated neuroplasticity, synaptic plasticity, might be important here. And we found that BDNF was playing some role that was just like Neuron-glial 3 that seemed to be important beyond its role as a mitogen, something else that BDNF must be doing, because targeting BDNF to track B signaling in gliomas is profoundly therapeutic in preclinical models. And so, we wanted to understand what was going on. And we find here that indeed, if we perfuse BDNF over a xenografted hippocampal slice, that the tumor EPSCs are much larger in the presence of BDNF. There's a greater amplitude, and that this depends upon the glioma expression of the BDNF receptor TrkB. If we CRISPR delete the NTRK2 gene encoding TrkB, then there's no effect of BDNF on the amplitude of the current. And just in the interest of time, summarizing these findings, what we discovered was that BDNF to TrkB signaling in the glioma cell results in increased AMPA receptor trafficking to the postsynaptic membrane, just as it does in healthy neurons. Which augments the strength of this synapse. BDNF to TrkB signaling also increases the number of synapses that form. So in summary, what I'm telling you is that gliomas are integrating into neural circuits. They are doing this synaptically through bona fide neuron to glioma synapses that are then elaborated and amplified through mechanisms of adaptive synaptic plasticity. In the interest of time, I'll just summarize that in addition to that, those synaptic currents, there are also potassium-evoked currents in gliomas that also get amplified, in this case, amplified through a tumor-to-tumor gap junctional coupling that occurs. And that's a lot of different ways that tumors are both undergoing membrane depolarization and modulating the degree of membrane depolarization. And, as we know, membrane depolarization is a very metabolically expensive thing to do. Tumors have to, after depolarization, repolarize in an in an ATP-dependent way. There must be something advantageous for the cancer cell if it's undergoing these frequent and robust membrane depolarizations. And it makes sense that there might be. We know that during brain development, that membrane depolarization of neural stem and precursor cells in the germinal zones is really important for appropriate corticogenesis. And so we wondered whether the tumor cells were taking advantage of some sort of voltage-dependent signaling pathway. And to test this idea that membrane depolarization itself is what is important, or one of the major things that is important to the cancer cell, we again turn to optogenetics, but this time using the blue light sensitive cation channel rhodopsin 2 expressed not in the microenvironmental neurons, but rather in the tumor cells themselves. And in this way, we can use blue light to depolarize the glioma cells, and we can do this in vivo. And what we find is that indeed, just optogenetic depolarization of the glioma cells increases the proliferation rate of the tumor. Now, it's not just that the tumors are depolarizing, but they're modulating the strength of that depolarizing current. They're modulating the amplitude of it. And so we can also use optogenetics to test the idea that a larger magnitude current might differentially promote proliferation of the cancer cell. So just by altering the pulse width of the light that we deliver, we can cause either a medium-sized depolarization or a larger depolarization in the tumor cell. And when we do that experiment, we find that indeed there is a differential effect on glioma proliferation rate of larger magnitude depolarizing currents. Conversely, we can reduce the neuron to glioma electrochemical interactions using multiple strategies. Here I'm showing you genetic blockade of the AMPA-dependent current through a dominant negative GLUA2 subunit expression in the tumor cell, but there are anti-seizure medicines and other pharmacological strategies that also can decrease this kind of signaling. And there is a start growth inhibitory effect of that. We can visualize these electrical currents using genetically encoded calcium indicators expressed in the tumor cell. And here I'm showing you GCAMP6S imaging in a diffuse intrinsic pontine glioma. And I think this particular calcium imaging really underscores the fairly startling new understanding that these cancers are electrically active tissues. And that is not how we have been approaching them, either from the perspective of understanding or treating them. And so now as a field, we really need to understand in great detail the mechanisms of this malignant circuit assembly, plasticity, and evolution over the disease course, to understand the granular details of these voltage-sensitive mechanisms of proliferation. And I'll just suggest that, in doing so, we're going to glean important insights for normal neural development and plasticity viewed through the magnifying lens of these glial cancers. In summary, work from my lab, but now from many others, demonstrates that malignant gliomas are really forming a network. They're forming a network integrated into the brain circuitry, and that is central to driving pathogenesis. Not only does neuronal activity drive glioma proliferation and growth through both activity-dependent paracrine factors, but also through neuron to glioma synapses, of which there are multiple types, it turns out. But the tumors in turn increase the excitability of the neurons, contributing to glioma-associated seizures, and also functionally remodel neural circuits to increase the inputs into the tumor. As we begin to understand these interactions, a number of potential therapeutic applications become clear. We may be able to target neuroligin-3 cleavage and or binding, and I've just completed a phase 1 safety trial looking at ADAM10 inhibitors in pediatric high-grade gliomas. We may be able to target AMPA receptor signaling, and there are anti-epileptic drugs that do this. Glutamate reuptake, neurotrophin signaling, synaptogenic factors, potassium channels, gap junctions, and in some subtypes of tumors, GABAergic signaling. And I'll just end with the idea that, targeting these neuroscience-inspired targets might be really important, not only for directly treating the tumors, but also they may synergize with immuno-oncology strategies. And this is the topic of a different talk, but together with Crystal Makel, who's left NIH and is now at Stanford with us, we've been leading a GD2 CAR T-cell trial for DIPG, in which we've had some absolutely remarkable responses. And this is a complete and durable response in one child. But in most cases, we're seeing less complete and less durable responses. And part of why that's true is that this is such an aggressive, fast-growing cancer that one can imagine the CAR T-cells just can't keep up. And so one question is, could targeting these neuron cancer interactions slow tumor growth sufficiently to help the CAR T cells outpace the tumor growth? And I think in another way, we can think about how neuronal mechanisms and neurotransmitter mechanisms might be modulating the tumor immune microenvironment in ways that counter CAR T cell efficacy and could be modulated to make them more amenable to effective CAR T cell therapy in this context. So much work to come on the intersection between neuroscience, immunology, and cancer biology. And I think that the mechanistic parallel is evident in normal neuron-glial interactions at play in adaptive myelination, and malignant neuron-glioma interactions really underscores the extent to which these cancers are simply hijacking mechanisms of neural development and plasticity, and really demands that we approach these tumors from a neuroscience perspective. So thank you so much for your attention. I hope there's still a minute left for questions. Many people to thank, including the people in the lab, past and present, critically important collaborators, our funding sources, especially our NIH funding, and patients and families whose donation of tumor tissue enables all of the work that we do. Thanks so much for your attention.
Thank you so much, Michelle. That was just such a magnificent and inspiring body of work that you presented. I just want to, we don't have much time, I just want to quickly see if Dr. Volkov has a question. Nora, unmute, you can unmute.
Yes, I have a fantastic presentation. And my question, though, and it was not clear in my brain, I mean, certainly the neurons seem to be driving the connections with the oligodendroglial cells, but once you create this malignant process, does the malignant oligodendroglial cells feedback back into the neurons, amplifying that cascade that stimulates them? Is it bidirectional?
Yes, it is bidirectional. The electrical signaling is only from neuron to glioma, but then there's paracrine signaling, and many mechanisms have been described. I think the most compelling are secretion of synaptogenic factors like thrombospondin 1 and glypican-3 that increase the excitability of the neurons and make them much more active, but also functionally remodels the inputs into the tumor so that a wider swath, for example, of cortex will be stimulating the tumor than might be otherwise activated during a particular task. And this was really beautifully shown using intraoperative electrocorticography by Sean Hervey-Jumper at UCSF, found that during language tasks, expressive language tasks, patients with GBM had much more cortical area activated when the tumors were infiltrating the left hemisphere than you would normally have in expressive language areas. So there's functional remodeling as well as increased excitability.
Thanks very much. It's a wonderful work.
Thank you.
I need to remind everyone that the CME code is 50107, and I'm going to ask a question. I just sneak one in. I wonder if you could tell us a little more about the dynamics, and I guess I mean time-wise, of how neuronal activity is inducing the number of oligodendrocytes, and sort of is it reversible? Because you showed this in related to substance use disorder, and oftentimes we think about recovery and what are the mechanisms involved in recovery. So I was wondering, what's the time course, and is this reversible?
Yeah, so the time course is very quick. We see the proliferative response essentially immediately, and then the oligodendrocytes are generated over the next several days, and we see the functional effects within a couple of weeks. But myelin plasticity is also bidirectional. So decreasing activity within a circuit has been shown to decrease the myelination of that circuit. And so there are several mechanisms that have been described for this. And there may be myelin phagocytosis by other glial cells, for example. So there is a sort of bidirectionality to the plasticity. And in recovery, decreasing the activity within, the reward circuitry could reverse those changes. But that's something that needs to be studied.
Great. I have a question from an audience member. Does neuroligin 3 play a role in the development of glioblastoma multiforme, an adult neoplasm?
Yes. So yes, it does. Neuroligin 3 strongly promotes adult GBM growth and progression, and in the absence of neuroligin 3, tumors don't form particularly well.
Okay, great. There's another question. To what extent do neuroligin 3 and BDNF operate at a local level compared to synapse-mediated modulation of OPCs? Do BDNF or neuroligin 3 operate as diffuse cues, or are they acting in a way to promote selective axonal myelination?
Yeah, this is a great question. So they function locally and in a circuit-specific way, but the question remains, locally, but we know that the more it is the active axons that get myelinated, and that signaling isn't that precise. So how do we know, how does the OPC know, or the generated oligodendrocyte know which axons to wrap? Are there, axonal cell membrane proteins that are expressed? We don't know.
Okay, I think we're past time, and I don't see any questions from the audience. So I think we can wrap up. I want to again thank our speaker. That was terrific. And if you have any questions, you can email her, I'm sure.